Hi friends, welcome back to my channel Anand Science Academy. Let's continue the chapter Principles of Inheritance and Variation. In this video, let's learn the topic Incomplete Dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 progeny had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, or otherwise known as snapdragon or antirrhinum species, is a good example to understand incomplete dominance. In a cross between true breeding red flowered and true breeding white flowered plants, the F1 was pink. Let's see how to work out this cross. First, let us take the parents, the phenotype red and white, the two contrasting traits of the parents, and the genotype of the parents are capital R for red, small r for white. And let's see the gametes produced by them. Red plants produced gametes with capital R and the white plants produced gametes with small r. When they are hybridized, in the F1 generation, the plants produced pink colored flowers. The phenotype of the F1 plants are pink colored flowers and the genotype is capital R, small r. When these F1 plants were self-pollinated or self-crossed, let's see the genotypes and phenotypes of the plants produced in the F2 generation. The gametes and the Punnett square or checkerboard to work out this cross. Here you can see red, pink and white colored flowers are produced in the ratio of 1:2:1. This phenotypic ratio is a deviation from Mendelian monohybrid cross where it was 3:1. Here 1:2:1. If you see the genotype, that ratio is also here 1:2:1. The following diagram is given in our NCERT reader to represent this incomplete dominance. This can also be used. Here one thing to be noted, in this incomplete dominance pattern of inheritance, the phenotypic ratio and genotypic ratio both are same, 1:2:1. This is a very important point to note. Once again, let's understand this clearly. When F1 was self-pollinated, the F2 resulted in the following ratio: 1:2:1. 25% red, 50% pink and 25% white. Here, the genotype ratios were exactly as we would expect in any Mendelian monohybrid cross, that is 1:2:1. But the phenotype ratios had changed from the 3:1 dominant recessive ratio. Here, both genotype and phenotype ratios are 1:2:1. This has to be noted once again. What happened was that capital R was not completely dominant over small r, and this made it possible to distinguish capital R, small r as pink from capital R red and small r white. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles they exist. Now, these two alleles need not always be identical as in a heterozygote. One of them may be different due to some changes that it has undergone, which modifies the information that a particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now, there are two copies of this gene, the two allelic forms. Let's assume that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for the production of the normal or less efficient enzyme. Or otherwise, a non-functional enzyme it may produce, or no enzyme at all. That means the modified allele may produce normal enzyme or less efficient enzyme. It may produce non-functional enzyme, or otherwise it may not produce the enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele. That is, it will produce the same phenotype or trait. That results in the transformation of substrate S. Such equivalent allele pairs are very common. But if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected, as in the second and third case. The phenotype or trait will only be dependent on the functioning of the unmodified allele. The unmodified functional allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Hope now you are clear with what is incomplete dominance, in which plant it is observed, and how to work out the cross, and what are the phenotypic and genotypic ratios of this cross and how it deviates from Mendelian ratios. Let's continue the chapter in the next video.